see that okay? My sort of title slide here. Okay,
Yes.
thanks, Christy. Yeah, thank you very much for the opportunity to speak. Yeah, happy to do that. Yeah, excited to just communicate some of the different projects that my group is working on. The title of this talk is Increasing Capacity for Invasive Species Detection in Hawaii. So there's a lot of different efforts going on in Hawaii. And so I certainly don't claim that this talk's going to cover all of them. This is just going to share, some of the different projects that my group is working on and folks we're collaborating with. And I fully recognize that there's a whole lot of other really interesting, innovative stuff happening around the state. So I'll go ahead and get right into it if I can. So lots of folks to acknowledge, colleagues and collaborators, students, many people, and because I'm going to be talking about a number of different projects, also a lot of different funders involved here. So I just want to take a moment to acknowledge all these folks and thank them. And yeah, okay. So I will go on. A little bit of introduction, if you're not familiar with myself. So I'm talking to you from here, rainy Hilo. It's a wonderful rainy day here at UH Hilo. I'm in my office, and we've got some great programs here for any students who are interested in coming out here. So I'm the chair of the Department of Geography and Environmental Science. We've got a great department, lots of field work. These photos were all taken pre-COVID. We are masked up now. And also affiliated with the TCBES program. This is a master's degree program, Tropical Conservation Biology, Environmental Science. Really excellent program, and a number of the folks on here are affiliated with that program as well. I also run the SDAV lab, the Spatial Data Analysis and Visualization Lab. And our mission is to apply geospatial tools to environmental problems of local significance, and then to try to disseminate those tools and knowledge to the larger island community through education outreach. And we work on a lot of different things. We work on helping civil defense and USGS map lava flows. We are doing a big project right now for Hawaii County planning, trying to map shoreline change across the whole Big Island. And we also do a lot of work with invasive species. And so that's what I'll be talking about today. We've got a great group in the lab. This gentleman, he's recently taken a new job. So Tim O'Sullivan is moving on. And so if you're interested, there'll be a job posting coming up pretty soon for, he's irreplaceable, but some kind of person to replace Timo. So if you could handle working for me, which I don't know if too many people would stomach that. But we've got a great group in the lab and do a lot of interesting things. In preparing for this talk, I was, I came across this paper, which was published last year, Technology Innovation, Advancing Capacities for the Early Detection of and Rapid Response to Invasive Species. It's an interesting paper, and it hit on a number of major points about technology advancements, information inputs, the availability of information, and then different types of ways of going about detection, identification, and then different types of response technologies and mechanical, biocontrol, and chemical. So I thought it was an interesting talk to, or interesting paper in reflecting putting this talk together. So I'll be touching on a few of these different components. And a good one to start with, and a good sort of case study example about getting enough information to make management decisions and respond to disturbances in our forests is rapid ohia death. And yeah, I've talked a lot about this in prior talks. This is consuming a lot of the efforts of my lab the last couple of years. But I imagine everybody on the talk here is familiar with ROD, but just quite quickly, rapid ohia death, that's an expression, or it's a characterization of the set of visible symptoms that infected ohia trees go through. They rapidly brown and die out and lose their leaves, and it's caused by two fungal pathogens. It's really problematic here on Big Island and as well as on Kauai, and luckily not as much of an impact on the other islands so far. And hopefully we can keep it that way. But a lot of what my lab's group has been doing is a lot of detection work for this. And I wanted to just bring up this case. This is from La Poille Forest Reserve here on Big Island. A couple years ago, unfortunately, there was an outbreak in this beautiful mature ohia forest. And as part of the response, Big Island Invasive Species Committee did a survey using their Mavic 2 drone, which is this little drone, can fit in a backpack, great to hike through the forest, really portable. And in this forest, which is a lot of tall canopy, it's difficult to operate a drone and fall within the FAA rules about visible line of sight. But they did a great job. They mapped 100 acres. And from that, we were able to get really nice imagery that my lab can use to, identify suspect trees, ohia trees infected with the fungal pathogens responsible for Rhabdohia. But 100 acres is not a really big area to cover. And so, but we can, see these suspects and we can map them out. Because we're interested in landscape scale changes and detection, we've also worked with David Okita with funding from the Ohio Volcanoes National Park and a lot of innovation from Russell at R&R Welding to generate this helicopter manned aviation rig for doing imaging. And that allows us to get basically drone quality imagery, but covering a lot more ground. And so instead of 100 acres, we can fly 9,000 acres, and that 100 acres patch is just in the middle right there. And so we can really start to see, how these things are moving across the landscape and better characterize these patterns. But it's still, that's only a portion of the island. And so, I'm greedy. I want more information. I want more imagery, more coverage. And so if we jump up to satellites, there's a set of satellites that have, less than 50 centimeter resolution imagery. This WorldView 3's got about 30 centimeters. And that allows us to, and it also has additional wavelengths that are helpful for doing identification work. But this gives you a sense about that scale, right? So that WorldView 2 image we're looking at from July, that's covering 125,000 acres. And so, much larger coverage. And so if we look at the, the suspect rod trees based on some work my lab's done, we can see, again, you're getting a larger shot of what's happening in the landscape. So this imagery, and this is what it looks like, this imagery is appropriate for detecting suspect ohia rod trees. If you already are somewhat familiar with that forest, it is not appropriate for trying to find cryptic small individual Myconia plants. And I know Myconia is going to be the topic for a couple of additional talks later on here. And so, there's this interplay between the spatial resolution and the spectrum, basically the overall resolution of the imagery and what it's able to do. And unfortunately, these large satellite images, which cover lots and lots of area, don't have the spatial resolution to detect some of these invasive plants that we care about. But we can see them with helicopter imagery or imagery from drones. And this is a drone image right here. I'm not sure how clear it's coming across, but it's got a lot of Myconia in here, including some Myconia in this little covered patch there that you may not have seen. When we fly these images, as anybody who does any kind of drone mapping knows, you end up with hundreds or thousands or tens of thousands of images. And so manually going through those images can become very time consuming and costly. And people get tired and such. And so one of the questions that we're wrestling with is, can humans or computers do a better job of detecting Myconia from aerial imagery? And it says Myconia there, but you could substitute, any kind of invasive plant for Myconia, right? It's the same kind of idea. And so this is a paper that Roberto Rodriguez, UH Manoa, now at USDA, recently published with James Leary and myself and some other folks. And basically did kind of a head-to-head competition of human analysts versus a computer using an artificial intelligence algorithm that we generated. Did this with some imagery from Big Island. And again, it's taking advantage of artificial intelligence. And basically what we're trying to do is train the computer to be able to detect our target of interest through eventually sort of deep convolutional neural networks, which is a subset of artificial intelligence. So artificial intelligence, machine learning, then into deep learning. And really what we're interested in doing is using computer vision to be able to do object detection, right? So we want to be able to do object detection within an image. Is the object that we care about present or not? And so that's what this paper was about. Again, comparing humans. And so we had human analysts, and maybe some of the people in this talk participate in those humans analyst trials. And if you did, thank you very much. But the humans were basically looking at images and scanning the images and trying to identify Myconia targets. And there were 3 different image speeds that they were, so it was kind of fast and then slow. And so presumably if you look at an image slowly, you have more time to find targets. If you don't have as much time to look at that image, you're going to find less targets, right? And then we compared that to a deep neural network, convolutional neural network algorithm that we created, which is basically you feed the photos to a computer, and then the computer will scan all those photos and do its best job it can to find the targets of interest. And then we compared the results from those guys. And so this is an example of a situation. Those green dots, if you see there, those are places where the human analyst said, oh, I think there's a plant here. This red dot, the human analyst put a dot here and said, I thought there was a Myconia plant there. There's actually not a Myconia plant there. And then this one, they missed it. It's a little bit hard to see here, but they missed a plant. So this is just an example of sort of the data that we had to work with. And this is, it's a little bit of a busy figure, but basically this green bar here, this says the recall, and recall is defined as basically of all the Myconia plants that were present in the image, what percentage of those Myconia plants were successfully detected by, in this case, the human. And the green bar is the human operating at the lowest speed, the slowest speed. They had the most time to look at the image. And then the middle speed, a little bit faster, they had less time to look at it. And then at the fastest speed, they had even less time to look at the image. And so these 3 bars, first 3 bars here are for the human. And what we see are, as the amount of time they have to look at the image decreases, their performance goes down. And so at the slower speed, they could see about 75% or detect 75% of the Myconia. At the fastest speed, about 50% or so. So these first 3 bars are for the human. This pink one is for the computer, right? This sort of DNN algorithm, this automatic system that you just feed it. You feed the computer the images and it will spit out what it thinks where the Myconia are. And it did better, it did about 83%. So in general, this detection algorithm, it outperformed the human analyst, and it also was a hell of a lot faster. So it was more than 32 times faster than the fastest scrolling speed. And if we had even a bigger, beefier computer, it would be even faster. So in this case, the outcome in the Paul Bunyan test, right, a man against the machine, right, the machine won. The machine did it faster, and it did it better. And our algorithm is only going to get better as we keep feeding it more and more training information and keep training it, making it better. We've developed these algorithms for coconut trees, for pandanus, for different stages of rod. But training these models is very time consuming. There's a lot of work that goes into it. And so we're interested in potentially leveraging existing vegetation detection algorithms to more quickly create algorithms for new species. So if we've already done the work to make one for Myconia, can we use that information to help us do a better one for guinea grass? And I've got a graduate student who's working on this question. And she's also asking, are there things about particular plants that make them better or worse candidates for generating these detection algorithms? Is there something about tree that makes it a really good or a really difficult target? And so she was looking at some of these different feature measurements. I think I'm running a little short on time, so I'll probably go through this a little bit. But what she found was that contrast, which is basically how different is the target from its surrounding background in the image. Contrast had a nice strong correlation with model performance. So the more contrast there is, and it makes sense, the more different the target is from the background, the better off that model's gonna work. And texture, which is a sort of another measure, also had some sort of weak correlation. And tree, this is Erica pointing sadly at a tree in Hawaii Volcanoes National Park. It's another candidate that we're looking at. And there's no shortage of potential candidates. This is some imagery we collected with a helicopter over Maui. I don't know if anybody knows what this might be. And again, I'm short on time. You may be saying to yourself, I think that's Himalayan ginger. And this is a photo, credit to Forrest and Kim Starr. Thank you very much. And indeed, this is a Himalayan ginger. So this is a mapping project for Nature Conservancy on the Waikamoy Preserve, and interested in mapping out the ginger in this area. She also did some stuff with cross-species transfer learning. I don't know that I have time to talk about that. Other than when species are more similar to each other, we've got a better chance of kind of bootstrapping existing models. When the species are very different from each other, there's less opportunity for that bootstrapping. And we've got a question about how to best share these detection algorithms with the wider community, right? We're creating these algorithms and we want people to be able to use them, but it requires a lot of fancy computers and powerful GPUs and all this stuff that is maybe not easily available computational resources for everybody. So we're trying to work this out, and we've got this idea. We just haven't been able to get it together. So if anybody wants to help work on this, I'd be more than happy to talk to you. But basically, the idea is that an agency, a group, flies an area they're interested in, generates a whole bunch of photos, uploads those photos to the cloud, right? Some kind of a portal, some kind of website. They upload the photos. The processing occurs in there. They say, I want you to look for Miconia and guinea grass and Australian tree fern or whatever the possible algorithms are. And then it'll kick out basically a text file or a GIS file of those potential targets, along with the score. And the score is the confidence in how highly the algorithm regards that it thinks it is Miconia, for example. So I want to build this, but we don't really have the resources to do this yet. So if anybody's interested in talking to me about this, happy to chat. And the last thing I'm going to talk about is some biocontrol efforts with Tracy Johnson, focusing on strawberry guava and deployment of Tectococcus ovitus, TO. So we're working out systems to do this via drone. This is an early prototype where we do singles. We're trying to release, being able to carry dozens at a time and release these via drones. We're also thinking about doing this, trying to do this via helicopter. And an interesting part of this is to monitor how well this biocontrol works. And so the Tectococcus ovitus, it generates these galls. You can see in these blue circles, there are these little galls. And this is a photo taken from a drone, and the photo's so good, you can see the individual galls on the individual strawberry guava leaves, which I think is awesome. Right, and so this is the drone photo, but you zoom way in, you can see these individual leaves and the galls. And this is a map of one of Tracy's earlier inoculation sites in the middle here. And we did this sort of real high resolution photo mapping work across this, this sort of star pattern. Every circle, there's a place we took a photo. And if those circles are red, it means there were no galls detected. And if it's sort of this bluish green, it means indeed galls were detected. And so, and then the size of the circle is the percentage of strawberry guava in the photo. So that's how to kind of interpret this graph here. And so you can see that, okay, there's a portion of the landscape. And again, the original inoculation site was right in the middle of that star, right? And so there's this sort of part of the landscape where we're getting these, galls, right? But there's another part where we're not getting the galls. And we've been talking to Tracy about this, and he very much thinks, wind is playing a role in this kind of a thing. So this is one, these are some of the examples of things that my group's working on. And I know I'm kind of taking some time. So I think that's the end here. So thank you all very much for your attention. And yeah, really happy to have the opportunity to present to you guys. So mahalo.
Thank you, Ryan. Very good presentation. And after this, we have a presentation from Christy Martin. Christy Martin coordinates a coordinated group of alien pest species called CGAPs. And one of the major work of CGAPs with the leadership of Christy Martin is to jointly work with HISC on the HISC CGAP 2025 strategic plan for Hawaii biosecurity. Christy, whenever you are ready.
All right, thank you so much, Roshan.
You're welcome.
Let me see, there we go. Does that work for everybody?
Yes.
All right. Well, thanks so much. I wanted to talk a little bit about some of the tools that have been developed in the past to identify invasive plants and how we're trying to use them to prevent the next Myconia from being imported and becoming established and causing harm. So I will move through this pretty quickly, and I have to apologize. I always include definitions and a little bit of natural history in there, so you folks can zone out for a little while if you need to. I always assume that there's at least one person in my group that doesn't fully understand why we're concerned. So, of course, we're isolated in the beginning, right? The Hawaiian Islands is an archipelago, and the islands themselves have been in existence for more than 70 million years. There has always been a landing spot in the middle of the Pacific during that time, maybe not the same exact island, but as the Pacific plate moves and new islands are formed, there's been landing spots here, but we've never been closer to any other large land mass. And so this means we've been surrounded by the Pacific Ocean, which forms sort of a moat around Hawaii. And of course, we teach the kids in school about the 3 Ws, how life arrived here, starting with plants like seeds, spores, insects, and even spiders arrived on the wind. A few birds flew or blown off course, and maybe in them or stuck to their feathers were more seeds. And of course, some seeds floated here on that 3rd W, the water, ocean currents and waves. And of course, ocean currents carried more and more plants that we know of now, thanks to a lot of the work John Price is doing. But we know also that they carried larval fish, eggs, even some invertebrates and algae and freshwater fish. And so things were able to arrive over time, become established, and some of the really lucky ones were able to form successive generations. And so what that brings us to is through isolation, a wide variety of habitats in Hawaii, lots of time and small changes in those species over time as successive generations came along, we came out with nearly 20,000 native species, and that's terrestrial and aquatic. And so native species, when we talk about that, these are the plants and animals that arrived at a location without the help of our jets and our super tankers and all of the descendants from those colonists. And so moving forward on our timeline, Polynesian voyagers arrived, and through their boats, they were able to bring the first non-native species. We used to call them alien. That's become a really disrespectful term these days. No disrespect meant. It's just simply a way to describe how things arrived, hitching a ride or being carried by people. And some examples include taro and even Polynesian rats. Moving forward on the timeline, when we got put onto the world map by Captain Cook, we had an immediate influx of new species carried by people, some purposely and some accidentally. And we really start seeing and documenting the effects of rapid introductions and harm caused by some of those species. Some of them were invasive, like those goats. You folks on the Big Island are still seeing the descendants of Captain Vancouver's gift to the king. And so today, or at least pre-COVID, we have a lot of residents and a lot of visitors rising up again. And we have a lot of introduced species that have become established. We never had any land or freshwater reptiles. Now we have at least 40. Hawaii Department of Agriculture estimates that there's at least 20 insects per year that become established. And I always point to a really excellent piece of work that Department of Agriculture did at Kahului, where they looked and they super staffed the airport to look in those conveyances arriving by air. And they were finding an average of 1 new plant, disease, or invertebrate arriving each day as an average. And that's Kahului, that doesn't receive a whole lot of incoming cargo. Just think about what comes in at Honolulu, either at the harbor or at the airport. So we know that number is huge and high. The question then becomes, well, which ones become established and escape? We don't know the answer. For the marine and brackish water environment, we have at least 420 new ones. And for plants, that number should not surprise you when you look around outside in any of our daily lives. You see lots of new plant species. Bishop Museum estimates that at least 10,000 have been introduced, and a back of the napkin estimate done by Cliff Smith and his team find that at least 200 of those are causing true ecosystem harm. I argue that that number is really quite a bit higher at this point. Now, if you're bummed out by now, I have to remind folks that the glass is still half full. There was some work done that estimated that about 50% of native habitat remains, and that amount is actually higher and more healthy in the oceans. But it is threatened by invasive species and climate change, and of course, those synergies between climate change and the effects on those invasives. So, should we try and keep all the non-native species out? Well, no, that's really unrealistic as well. But we should focus on the invasive species. And we've got some of the tools these days to figure out which ones are invasive. And people always ask me, well, what do you call invasive? Well, we use the federal definition. And that is the non-native species whose introduction causes or is likely to cause economic, environmental harm or harm to human health. And so how do we figure out which plants, just as one subset of the problems, how do we figure out which plants are going to be invasive? Well, that's where the weed risk assessment came in. And I've got just a short timeline of some of the work that went into it. Really, it's just a huge body of work, people working off of other ideas and developing and testing. And so just very briefly, in 1994, the weed risk assessment was actually developed in Australia. It's just 49 questions about the life history and performance of a plant. And it functions like a background check, pre-Google. In 95, that was adapted and adopted for use in New Zealand. It was also tested there. In 1998, our Dr. Kurt Daler at the University of Hawaii Manoa adapted it for use in Hawaii and gave it a new name, the Hawaii Pacific Weed Risk Assessment. So now we've got that long acronym, HPWRA. And look, that's when that executive order, that federal executive order in 1999 came in that directs federal agencies and those using federal funds to not authorize, fund, or carry out actions it believes causes or promotes the introduction or spread of invasive species. So that got some folks to thinking, particularly the federally funded Ka'ulani Urban Forestry Program. They said, wow, we use federal funds to promote tree planting. And so this caused Ka'ulani to invest in the testing of this system for Hawaii and found that it was more than 90% accurate at flagging plants that will become invasive. And so in 2005, really through the present, it was transitioned to the Hawaii Invasive Species Council. Chuck's going to be talking to you later today. Chuck is the weed risk screener. We're so lucky to have him. And honestly, this service is a free service and it's for information purposes. So people can make better importation and planting decisions. Just a little bit more about it, you know, those 49 questions, it gives it a score. It can get a negative score, so you can reduce the risk if you answer in a certain way, but it uses published information and it correctly flags 95% of the high risk plants. We have a website if folks want to take a look at it and the plants that have been screened. That's plantpono.org. But moving forward in my story, you know, now that we have a tool and it's published and it's easy to use, these are some screenshots of some of the scores and how it's viewed on Plant Pono. Myconia came out high risk, 14, because of its history elsewhere, and its traits. Plumeria is low risk because of its history and traits. So we have all of that. Well, okay, so how can we use this to form better regulations and keep some new invasive plants from being imported? Well, in short, it's legal to import 99.9999% of plants living on Earth, and there's nearly 400,000 species of plants. So there's lots of them out there. It's very difficult to to restrict the importation of even the known invasive plants because of how the regulations are set up. And I'm going to be very simplistic in my definition, and very happy to be corrected, but sorry, this is my simple explanation. So there are federal regulations. One of them is the federal noxious weed regulations and the list. It restricts the importation of a set of plants from foreign sources, primarily. But many of these species on this list are actually already present in Hawaii. It's tough to get plants on this list for lots of reasons. Another regulation is that there's a state list from our Hawaii Department of Agriculture for restricting imported plants from domestic sources. These are, from other states. But the list and the current rules are for plants that carry pests, not for plants that are themselves invasive or harmful. So we have a gap, and we're called C gaps for a reason. So we see this gap in our ability to restrict invasive plants. However, there's another problem. So a state could restrict the importation of a particular plant. We find an invasive plant out there, it's really harmful and invasive. And let's say we're able to get it on a state Hawaii Department of Agriculture list. This only restricts it from domestic importation. You can still import that same plant from foreign sources. So you need to enact a regulation on the federal side as well. You might be saying at this point, wait, what? So here's my storytelling. And I mean absolutely no disrespect. This is my simple way of trying to walk us through what's going on. So the feds say we have the authority over which species are pests and which ones are trade commodities. That is their role. We are, by the way, we're not going to regulate it until the state regulates it first. The state has to prove that they're serious about this plant before we do anything about it. Totally makes sense. Okay, state of Hawaii says, at least until 2008, they said, we don't have the authority to restrict the importation of a plant unless it poses a risk of carrying a pest. So we can't list a plant just because it itself is invasive. That's what they said. So we went to the legislature with a team of partners, and the 2008 legislature passed this bill. And they said, you can now restrict the domestic importation of harmful plants. However you want to define harmful, you can restrict harmful plants. And you can also regulate their sale. And for good measure, by the way, all of the noxious weeds, which is not a restriction list, it is not to restrict their import until this law passed, they put all of those noxious weeds onto the restricted plant list in 2008. So now you can no longer import Myconia, which actually was great. That was a huge gap too. I didn't want to talk about that gap. In 2008, governor signed it into law. Great. So the state said then after that, we need to adopt rules for how harmful plants will be listed for import restriction. Absolutely. They just have the authority. They need rules to be able to say how this is going to be accomplished. So what happened there? Well, if we're going to enact rules, we may as well enact a list also, because rulemaking takes at least 2 years. So let's do some research and figure out if we can use the weed risk assessment to identify that list. So we contracted the former HPWRA weed risk screener, Shaheen Ansari, who got her PhD working for Kurt Daler to do this weed risk assessment. She put together a process for identifying new plants that are not present in Hawaii that could be the basis of this proposed list. So she gathered an initial list of over 1,000 invasive plants that were already being regulated by other states and nations. So from this list of other lists of plants that were already invasive and being regulated, she assessed whether or not they're in the trade, therefore likely to arrive. She then verified that each plant species was either not known to occur in Hawaii or are present in very limited numbers and locations. And then she screened all of them, hundreds of plants, using the weed risk assessment. Then that subset of plants, she gave us a list from that subset of 36 plants that scored high risk, not present, and we used it as a basis for that initial list of things to propose for restriction. We also went on to work with Chelsea Arnott, who's on this call. She was a grad student at NREM at the time, and her master's program was to spread awareness about this project with the local landscape and nursery industry. Chelsea also conducted an outreach. She conducted a survey to see if these plants were going to affect people's businesses, just to understand how regulatory actions would affect the industry. She then graduated and was hired as our planner. I was so excited to do that. She was able to continue. We also hired a legal expert, a lawyer. Catherine Stanaway was our legal fellow. And she consulted with Department of Agriculture, thanks Jonathan Hove for spending so much time with this, and other Department of Ag staff to propose to draft some proposed rules. And in February of 2019, we petitioned the Board of Agriculture to enter into rulemaking for these rules and this proposed list. The Board of Ag at that meeting voted to send the proposed draft rule to the list and the list to the subcommittee on plants. And so just so that folks know, that would be the entry into rulemaking. The subcommittee has to vote on whether or not to enter this into rulemaking to start the process. I don't have a graphic for what the rulemaking process is for Hawaii Department of Agriculture, but this is the one for the Board of Land and Natural Resources. It's a very similar process. There are multiple steps where there's public input into the process. And so I just wanted to say that that's kind of where we are right now. And a reminder that the rulemaking process can take at least a year, sometimes a lot longer. Hawaii Department of Agriculture is stretched really thin. They've got a whole bunch of new pests and their existing duties that they are already covering. So, you know, there's if you're going to ask me why it's taking so long, they've got a lot going on. But we are hopeful that sensible rulemaking like this project can be taken up so that we can prevent costly new invasive plants from arriving. And I just really appreciated this graphic that I borrowed and sort of modified. I feel like sometimes this is what policy work looks like. So I went over, but I just want to thank these people for their excellent work. We stand on the foundations that they build. So mahalo.
Thank you, Christy. So questions for Ryan and Christy? Is there? Anybody wants to ask question? So otherwise, I have one question to Ryan. So you take the aerial photo, and then looking at the photo in the computer, you can tell which species, multiple species, right?
So for some species, we have been able to generate algorithms that can automatically detect if that species is present or not. So there's a small number of species that we now have algorithms for. So that's correct.
Okay. So again, one more question. I'm a little bit unclear about what is algorithm. Sorry.
Okay.
Yeah.
So that word will just encompass sort of this black box. The idea that you have a bunch of photos and you basically give it to a computer program, and then the computer program uses the algorithm, and it basically uses edge effects and a bunch of other things that it's been able to learn that's what Myconia leaf looks like. A Myconia leaf looks like this. And the reason it knows a Myconia leaf looks like this is because we've given it a bunch of training data, and we've given it a bunch of time to sort of work with all this imagery and figure out what Myconia looks like. So the algorithm is basically a program that you feed it photos, it ingests those photos, and then it kicks out at the end coordinates of any potential Myconia that were present in those photos. So both the geographic coordinates, but then also a score. And that score is how confident the program was that it was Myconia or not. So you could say, show me all the Myconia that you were confident of at least 85%. And it would be a set of points would come out with geographic coordinates. And then those points, if you go to the forest, presumably you would, and nothing's changed, you would find Myconia at those points.
So it looks like it will take shape and size rather than color, hue and chromes, right?
It takes all of that information into account. And I think there was a question about, looking at longer wavelengths, infrared and such. The imagery that we've primarily been using for training is visible wavelength imagery. So it's red, green, blue. So there's not a so far we've been focused on visible wavelength imagery, just like you take with your cell phone or something, because the spatial resolution that we're able to get from these new cameras that are coming out are high enough that we're able to identify these species without needing to go into other wavelengths.
Okay, thank you. So I have another question. If we have something new species to detect, how does that work with the imagery stuff? Some new plant, not Myconia, not Ohia, or some new. Do you have to find out this all, or there will be computer planning, putting the shape and size and color and stuff?
So that's part of the challenge that my graduate student Erica is working on. So if it's a brand new species, typically it's not widespread enough for us to get all kinds of images to use for the training. Right? By the time there's enough, imagery available for us to build up a very robust training data set, it's probably too late because then it's spread out too much. So Erica is looking at, can we use very, very small imagery data sets to train these algorithms that have already been created for other plants that are kind of similar to these ones. And then another idea that we've also had is, if we know what the host country is for this plant that's come in, are there folks in those host countries that have drones, have imagery that they'd be willing to sort of share imagery that then we could use to do more rapid training?
Thank you. So there is a question about portal concept. So it says, could have any distributed computing. I love the portal concept. So I think the question is.
So it can go a bunch of different ways. So at Manoa, they've got a very high performance computational cluster. It's called MANA. And so we've used that for some things. And so that would seem to be a nice local resource that we could draw upon. But there could be distributed aspects to it as well. I'd be happy to talk different variations and what this might look like in the future. But because we actually tried to share one of our, I think it was the Rapid Ohia death one, with folks over on Kauai, and their existing computers just couldn't handle it, right? And so we're running into this problem where they just didn't have the computational infrastructure, but they've got a bunch of imagery they collect. And so how can we make this easy for folks? They've got the imagery, they just upload it, and then all this stuff happens in the cloud and then comes back to target. So that's the idea, to try to make it easy, because we want this to be used, right, and widely shared.
Thank you,
So-
Ryan. Yes, please.
I have a question. So what you're talking about right now, so the existing imagery, say where we had someone doing all the aerial reconnaissance for ROD on Maui, on West and East Maui, can that data be used for looking for, let's say, for Myconia?
So it depends on the resolution of the imagery, right? So if the resolution of the imagery is fine enough, yes, you could use that same exact imagery. If I don't know if you recall the example I gave for the satellite imagery, right? That satellite imagery, we can use that to find suspect rod trees, but I can't use it to find Myconia because the pixels are too big, right?
Okay.
So that's a limitation, depends on the imagery.
Thank you.
Yep.
Thank you. So there is a question. So it looks like it's for Christy. If we want to contribute observation to the HPWRA process, is there a format that we need the data in? Christy.
The best contact for that would be Chuck Chimera, and he has posted his information in there. And so Yeah, we're at the wonky office, and Chuck is at where the sort of the point of the arrow for weed risk and what's actually going on in the field for some of these pests.
There is one more question. How do we know when the rulemaking process has begun by SDOA? Will they reach out for public input?
Yeah, the good news is that rulemaking is a public process, and because there are multiple steps in the process, there are multiple public meetings, and public meetings are required by law to have notification. And the problem, though, is sometimes those meetings are scheduled just a week in advance. So you do need to either pay attention, or here's the better one. You can contact the Department of Agriculture to get on the list for notification for Subcommittee on Plants and Animals and the Board of Agriculture meetings. There's also another meeting, the Small Business Regulatory Review Board. That board makes sure that there's no impact to small businesses from regulatory actions. That also is a publicly noticed meeting. So the rulemaking process is long and onerous because it is a public process and we all have a voice.
Thank you. So there is another question. So list of 36 invasive species. Where do we get this? Department of Ag?
Dang, I'm going to see if it's posted on our website, but a very quick look through our website. We just reorganized it. I can't find it. So let me find it. I will post it to our website and then post it in the chat during this conference. So sorry about that, folks.
No problem. Thank you. Another question, I think it's still for Christy. Octopus tree and pencil cactus are sadly still being grown by Hawaii residents. How do we stop that?
Yeah, you get that rock rolling up the hill thing. It's really hard, especially for something that is so widespread. If one person controls it, what happens if their neighbor does not, or if it's just growing on foul land next to you? Kudos to the people that still try to point it out in a nice way and try to offer alternatives. At this point, though, octopus tree is so widespread that one of the very few options we may have will be to look for classical biological control, that is going back to the source range for octopus tree and finding that very close co-evolved parasite, plant pest, natural enemy that is only found on it, and see if we can reunite these old partners, because that's what slows the octopus tree down in its native range. That would require a lot of work, but honestly, we don't have anything better against octopus tree.
Thank you. So lots of questions, actually. There is 16 new questions. So another question, how to get more public awareness and retail market cooperation for pono planting in Hawaii?
Yeah. We need lots of help. The invasive species committees are some of our lead partners in this. They are working with on each island to try to raise awareness. On the Big Island and Kauai, they've done some particularly targeted work and have created pono endorsed businesses for nurseries that agree to a set of best management practices. They agree to be inspected also, not by a regulatory agency, but just spot check to see how they're doing. See if there's little fire ants, see if there's frogs, see if they're using weed risk assessment for their imports and things like that. So those businesses are posted also on the plantpono.org website. It's tough because we're pushing and trying to drive public demand as well as industry response. So it's really difficult to do that with $0 and just sort of, just shoe leather and hit and enter and stuff like that. So if anybody has any great ideas for how we can generate more attention on particularly those good actors and drive more people towards that voluntary program, that would be helpful.
Great. We have lots of questions, which is very good. So do we have any list specific to regions or by island? I think
I would say the best place to turn for that is the invasive species committees for incipient plants. Those are plants that are not widespread. And of course, they are niche groups. They try to focus on those early, just becoming established worst invaders. And a few plants that are causing, over and above damage, like Albizia along roadways. So that would be a great place to turn for island-specific high-risk species. And until we get better regulation or a more overarching outreach list, just for people's awareness, I would stick to the invasive species committees, and then just go to Plant Pono and see if a plant you're looking for is listed there as one of the high-risk plants. You can sort the list of plants that Chuck is screened by how it's screened out. So you can look at all of the invaders at once. You can look at all of the low-risk species at once. You can look for particular plants. And you can also look by, if you're a landscape architect, for example, and you need a ground cover that's low risk, you can also search by form and function for low risk plants.
Thank you, Christy. So we will skip our break, which is for 5 minutes, and we will go ahead with the Q&A session. So this would be a question for Ryan. What is the average cost per acre to acquire and process remote sensing imagery?
Well, that's an interesting question. So I don't know. Every place is a little bit different, but I can definitely tell you that once the acreages get above hundreds of acres, it very quickly becomes much more cost effective to use helicopters. And the helicopter fuel price may vary island to island, but so you can just take that into account. But we can typically map, if we're doing rod mapping, 4,000 to 6,000 acres an hour, something along those lines. And it also depends on how far you got to fly from the refueling location or such. So and then there's the image processing side. So there's the image acquisition part and the costs associated with that. And then there's the image processing part that comes afterwards. So yeah, I'm sorry, I don't know if that's helpful. I can't really pin down much deeper than that. But that's kind of ballpark.
Okay, thank you. So I have a question for Ryan. So you showed a imagery of strawberry guava, with the galls. So galls mean that had been damaged by the natural image, the scale insect, tectone, something. I forgot.
Yes, those galls are due to the TO, the scale insect that has been deployed as the biocontrol. Yes.
So that scale insect, does that scale insect kills the plant?
No, it doesn't kill the plant. And Tracy Johnson is the expert on this. It doesn't kill the plant, but it can really slow it down and kind of knock it back. So it basically weakens the plant and can slow the spread. But Tracy's the expert on that. Tracy Johnson, USDA. But so it doesn't immediately kill the plant. It just sort of slows it down.
Okay, there is still lots of information shared and question answered in the chat box. I will just move quickly ahead. If you still have lots of questions to Ryan and Christy, please feel free to put the question in the chat box or email Ryan and Christy. Let me see if there is any other. So yeah, it looks like there is lots of information. So maybe there is one question. Can the use of imagery be used to detect diseases in the plants by using different spectrum? So that's the question. Yeah.
Sure. Yeah. And the answer to that is yes. And we've actually published a paper trying to get at the early detection of rapid ohia death using hyperspectral data. Greg Asner's lab has done a fair amount of work on this. And basically, as you get out to longer wavelengths than what our eyes can see, there is some potential to start to see plant stress sooner than what we can see with our eyes. There's been a lot of work on this primarily in agricultural areas for nutrient deficiencies and early indicators of drought and other things. So the answer is yes. And it's somewhat plant specific, but in general, in the near infrared, there's a very simple, it's called a band ratio. It's normalized difference vegetation index, NDVI. So this has been used for more than 30 years trying to get at plant stress, and sometimes able to see that before your eyes can see it. And there are some very wavelength specific ones for specific types of plant diseases and for specific species of plants. And so the answer is yes, but you quickly go from the world of very easy to take photographs to thinking about image calibration and a bunch of other things that start to play a role. So the answer is yes. There are drone cameras and sensors that are sold that are either hyperspectral or multispectral sensors. We have some of these in our lab, and there's good potential for that, but it does get a little bit more complicated.
Okay, thank you. Thank you, Ryan, and thank you, Christy. Okay, welcome back again. Our next presentation or talk is from Nate Doobie. I just learned the last name. And he's a manager of Oahu Invasive Species Committee, and he will be talking about modeling Myconia distribution and spread in Upper Ko'olau watershed. Nate, whenever you're ready.
Thanks, Roshan. Good morning, everybody. My name's Nate. I'm the manager of OISC, and today I'm going to be talking about predicting the spread of Myconia using GIS modeling and the variety of ways that we use the information from the Myconia model here at OISC. So to start, I'll just go over a brief species description. I think most of you are familiar with Myconia calvescens, but for those of you who are not, it is one of the 100 worst invasive species in the world, according to the IUCN. It's a large tree in its native range, up to 15 meters tall. On Oahu, it caps out closer to 5 to 10 meters. The most distinctive characteristic of this species are the bright purple undersides of the large oval-shaped leaves. The tree has white to light pink inflorescence. The fruits are small, sweet, very attractive to birds. Birds are one of the main vectors of spread for this species. Myconia trees begin producing fruit as early as 4 years, and one plant can produce up to 9 million seeds each year. That's a really important reproductive characteristic to keep in mind as we go through the modeling. I wanted to include this picture just to emphasize the distinct underside of the large leaves. This is a photograph of former field staff after a successful Myconia control mission out in the field showing their trophies. But as you can see, they're very distinctive and very useful, as Ryan Peroy was discussing earlier, depending on the resolution of your imagery for identifying these species, whether out in the field, on the computer, from the helicopter. And when Myconia gets introduced to tropical and subtropical ecosystems, its reproductive capacity and its lack of natural predators in areas where it's not native make it a very prolific invader. So as you can see over here, get my laser pointer out. On the right side, all of these trunks on this photograph, this is all Myconia, and you can just see the erosive capacity. It forms monotypic stands. It shades out a lot of native species. What it does with its leaf morphology and its growth habit is it reduces the understory beneath the canopy and turns it to bare earth. And then in addition to the shading out capability of this species when it forms these monotypic stands, then the reproductive capacity kicks in. And as you can imagine, it quickly takes over large tracts of forest in its prime habitat. So with 9 million seeds per year, They're very small and very easily spread by birds and other animals when they eat the fruit, but we shouldn't ignore the human component. The seeds are also easily hidden in mud, and if you're not following proper decontamination protocols, humans can easily transport this seed and transport Myconia into new areas. So one way that we like to look toward the future is by looking at a case study of Tahiti, which has very similar habitat and ecosystem to Oahu. So Myconia was first introduced in 1937, and since it's overwhelmed over 2/3 of Tahiti's forests, is directly responsible for threatening 25% of their native forest species with extinction. These figures are a little bit old. I think they probably have increased since the time of this study, but those are the most recent figures that we have. Dense Myconia stands occur across approximately 80,000 hectares from sea level to 1,400 meters in elevation, with rainfall patterns similar to the Ko'olau range, and that's on in Tahiti. For comparison, the Ko'olau range is 40,469 hectares, and the highest peak, Konahua Nui, is 960 meters. So we're looking at very similar habitats here. And I just wanted to include these pictures from Tahiti. On the left, the two smaller pictures you can see on the top, this is a zoomed in picture of a monotypic Myconia stand. As you can see, at this level of canopy and level of growth in this particular stand, you can see no other plants growing in or around the stand of Myconia. You zoom out, the picture below shows the zoom out view, and you can see how rapidly it has pushed out quite a few of the other native forest species here in Tahiti, and these taller species are likely from before the Myconia invasion occurred. The Myconia, because it's so good at shading out, has reduced the recruitment for these individuals, and it's likely that when these taller trees die off, Myconia will take over and nothing else will grow. So in addition to its reproductive capabilities, Myconia is known for characteristics that promote erosion, and that leads to increased runoff and flooding. First, the pictures on the right, you can see that's within a monotypic Myconia stand in Tahiti. You can see the bare earth, not much else grows beneath the canopy. And the left picture is a photograph of one of our field staff pulling Myconia from an area that has had previous individual Myconias, and you can see the erosion starting in that area as well. So it's really the combination of the shallow root system and the leaf morphology that promote this erosion. Studies have shown that amounts of rainwater that are collected on the Myconia leaf surface are greater than most of its forest counterparts, and that the leaf morphology funnels rainwater in larger droplets with increased force to the bare earth. And this loosens a lot of the topsoil. And when you get a heavy rain event, of course, you're going to get increased sedimentation in streams. You're going to get reduced water going back into our aquifers here. And you're going to have to be dealing with a lot of nutrient-rich waters in the nearshore areas, as well as in the streams. And a lot of that is inhospitable to many of our native organisms in those habitats. One of the other things that I really want to highlight, in addition to the erosion, runoff, and flooding, is right now, you've probably seen in the news, rapid ohia death. We talked about it a little bit earlier, but on Oahu, if we were to lose our ohia trees, Myconia would be the most likely candidate to replace these trees. And when Myconia starts to get up into this native intact forest at the summit, you're going to see Myconia overtaking what used to be ohia habitat. Should rapid ohia death decimate our ohia populations, and should we allow Myconia to establish further in this upper native watershed? So it's a really big problem. OISC has been dealing with this since 2002, and it's our #1 target species and one of the 2 species that our organization was founded upon. So just a quick update on rod detections. Luckily, we've only found the less virulent strain of Ceratocystis huliohea. It should be noted we still are in the process of figuring out more about the Ceratocystis fungal pathogens that cause rapid ohia death. They're new to science, and we don't know a whole bunch, but we do know that the C. huliohea is much less virulent than its counterpart, Ceratocystis leucoohea. So far, luckily, we have no leucoohea detections on Oahu, but we do have 8 Juliohea detections. And as you can see across the map, some of them occur in the summit areas, some of them occur in the coast. And it would just be, it compounds the problem with ohia dying and showing symptoms of rapid ohia death and Myconia populations increasing across the island. So what do we do when we find a plant? Just a quick rundown. At OISC, we utilize a distinct buffering system and systematic survey techniques. We draw a 1,600 meter total buffer around the location of each plant, and that's based on seed dispersal distances. There's an inner 800 meter buffer, that's the green, and an outer 800 meter, that's the aerial. And on the right here, you can see when we get multiple plant detections, it turns into this globby polygon that represents our search area. So I do just want to mention that the seed dispersal distances and the buffering system are based on a lot of OISC data that shows that 99% of all immature Myconia plants are detected within 350 meters of a mature tree, and 99% are detected within 1,600 meters of the next closest tree. It's also based on the gut retention of the red-vented bulbul, as this is considered the most common disperser of Myconia on Oahu. So we took into account a variety of sources and based our modeling system and our buffering system off of these vector spread data points and off of some biological reproductive data points from the species itself. And then once we have our survey area, we have teams deployed to go out and survey and control any Myconia that they found. So our field staff conduct sweeps through the survey area using GPS and topo maps. They hike through dense vegetation, very steep terrain along compass bearings, through off-trail areas in the forest. They're usually able to hand pull the majority of Myconia plants relatively easily because of that shallow root system, but they do carry a little bit of herbicide in case we need to apply that to a stump for the large trees. And for any areas that they come across that are too steep, and any area within that aerial outer 800 meter buffer, we're going to use a helicopter and survey in a similar fashion, where we run elevational transects and use the helicopter's rotor wash to turn over the canopy of the Myconia and show us those bright purple undersides of their leaves for detection. So now we'll get to the Myconia model, that brief overview of Myconia. Thanks for those of you who are familiar for sitting through that. And what this Myconia spread model has done for us is it's allowed us to be more strategic in terms of deploying field staff to certain areas. It has helped us secure more funding, and it has helped us secure access to certain areas as well. So it's a very valuable tool for us. A couple of quick notes about the parameters. This model is very conservative. It projects each mature tree to only produce 20 viable seeds per year, rather than the 9 million that each tree is capable of. Plants begin producing seeds at age 4. The habitat is set as areas that receive 1,800 millimeters of rainfall or more a year and along streams. Myconia does like very wet environments, and that information is based on a lot of the data that's been collected through published sources about Myconia and its native range. Areas within our buffering system that do not meet the rainfall requirement are given a 50% survival rate. And that's because Myconia has been found on Oahu outside of these optimal rainfall areas. So it's kind of our best attempt to encompass all of the possible locations that Myconia could be. We also excluded slopes greater than 60 degrees, and that's based on internal OISC data from years of field observations. When we do find mature plants, we take note of the slope, and we've never found plants growing on slopes greater than 60 degrees. Impervious surfaces were also excluded, as Myconia can't grow on impervious surfaces, excuse me. So with that, the first thing I want to do is let a little animation run that shows the Myconia spread model. And this was originally done for a poster presentation maybe about 10 years ago, and then it was revamped for the World Conservation Congress, just as kind of an island-wide Myconia distribution model. And so what you'll see is what would happen if OS did not start controlling Myconia in 2002. And you can see how exponentially these populations grow, both outward spatially and in terms of density. And so this is a really helpful tool in showing funders that if we can't secure funding to continue the work that we've been doing for 2 decades, Myconia will inevitably spread into the summit regions of the Ko'olau and spread throughout the entire Ko'olau range, and would have some devastating consequences for our island ecologically and for a lot of the communities surrounding areas that have Myconia infestations. So these are some of the first images from that poster session, an original Hawaii Conservation Conference poster session. And this is year 0 in the back of Manoa. And if we let the model run for 10 years, this is what our model would project the area to look like in terms of Myconia coverage. So the model shows that after only 10 years, large monotypic stands are likely to form, making suppression and eradication impossible. The model it's not really intended to guide managers where to survey, but it's more to assist our organization with kind of explaining why Myconia is so detrimental. So the rapid spread of Myconia indicated in the model encompasses recreational and natural areas, which has further complications. Extensive stands of Myconia would also put nearshore waters at risk for increased runoff, and endanger the island's water supply because of the reduced amount of water going to our aquifers. And to give you folks just a general idea of where we conduct our Myconia work, this is a snapshot of our 2020 effort. It's a little different than most years. As you could guess, we had to mitigate some things because of COVID, but we were still able to get a sizable impact in our areas. And what I want to draw your attention to is the mature controlled right here and the immature controlled. So these are the areas where we really want to focus our efforts for our highest priority. So if we zoom in, what we'll have is two groupings, if I can get the animation to work. Hold on. Sorry. There we go. We'll have two groupings. The first one is going to be what we consider the Alawai watershed, and that's made up of Makiki, Manoa, Nu'uanu, some other watershed areas like Palolo, but we have yet to detect Myconia in Palolo, thankfully. So we're going to exclude that. And then we also have a windward grouping of watersheds that are high priority for us as well. So if we look at 2021 so far, you can see we've got a few more plant detections, and that's not uncommon because we do survey areas on a 3-year rotation just to try and catch any plants before they mature. So some of these areas are going to be different than 2020 just because we're repeating areas from 2018 rather than 2017. But for some of our areas that we are focusing on, what you'll see is a little bit increase in numbers, and that's really troublesome for us, especially this last one that we're going to add into the purple group. That's technically on the leeward side, but it's being fed by a windward population, and we'll get to that in just a few slides. But finding these 4 individual plants in the upper Waiava watershed presents a big warning sign for land managers on Oahu that Myconia is now starting to reach the summit areas of native intact forest. So what are we going to do about it? So we wrote for some more funding to focus some of our efforts on the Alawai because it is one of our highest priority areas. And you can see here, this is a distribution of The control points and the survey area starting in 2002 through 2020 of where the majority of our Myconia work takes us in the Alawai watershed. So it's important to note that these dots don't represent individual plants. There could be a number of plants that fall within the 10 meters of this of these dots. And it's also good to note that there are many layers of these dots year over year underneath. So it doesn't do a great job at showing just how much Myconia we've controlled there over the year, but it does give you a spatial indication of where we're focusing our efforts. So we plug that into the Myconia model, and this is what it spits out for us. The green layer represents impervious surfaces as a parameter. Myconia won't be growing on impervious surfaces. We started with 24 mature Myconia, and that's because that's how many we detected between 2018 and 2020 in this area. All except 2 of those, actually all of those now have been treated. At the year 5, the model estimates 3,541 plants with a 46% increase in species density. And at year 10, the model estimates 262,613 plants with a 3,400% increase in plant densities since year 0. So you can see the model working, and what it's spitting out should be sending red flags to all of the funders that were requesting money, all of our partners, just demonstrating that the work that we do is very, very important, and we need to continue acquiring funding. So I'm running out of time. I'm just going to go rapid fire real quick. Just a quick demonstration of our buffer spread. Luckily in 2020, we didn't really have an increase in new areas, but we did have an increase in areas where we found mature points, and that pushed our buffers up to the summit area. So you can see that in the top part of the map in Waiava, and in the bottom part of the map, a mature detected in Manoa pushed our buffer system up towards the summit. So if we switch over to the windward side, you can see that the 2 populations, those are the Waiava points that I'm talking about that were recently detected and treated earlier this year. And then we see our historic population that we believe is now feeding those Waiava populations on the windward side, just on the north slope of the middle ridge that splits the Ka'alaia watershed. So again, we do the same thing. This is just to show you the populations, the 2 over the summit on the leeward side and the known historic populations on the windward side. This is after year 5. And again, you can see those original populations, and this is after year 10. The model output indicates that from the 2 mature and 202 immature plants that we found so far this year in Ka'alaia, the population would balloon to 37,471 plants by year 7, 581,867 plants by year 10. Again, these are the locations of those original populations. So disparaging, we are focusing our efforts there, but it is starting to spread into the intact native watershed. I'll go quickly through this one. This is just another indication. This is a population that was over in Kahalu, and we used some modeling to show the same thing in order to secure access to this area and to get approval for our operations that we were trying to get through at that time. So again, you can see the current Myconia. You can see after 5 years and after 10 years, it just really blankets that whole area. So it should be noted that we do have most of our funding goes towards Myconia, a little over half, but most, and or I guess the majority. But we do have less funding compared to previous years. We do have the lowest field staff numbers since 2013. So with plants popping up in the summit areas, low capacity, low funding, we're looking at not a great formula for success in the future. So we are awaiting news from new funding sources. I will say I can't speak too much on it. I'm not sure if the press release is out yet, but we did secure some additional funding from one of these sources. So that's really exciting news for us. But we need to make sure that we continue getting enough funding to maintain our Myconia eradication efforts so that we can one day get to that eradication goal. And with that, I just want to thank all of our partners. We have a great partnership network and funding network, and we're really stoked to be working with all these great people. And if you folks have any questions, you can reach out to us. It's our website. We recently got a TikTok, so if you want to check out some funny TikTok videos of invasive species and things, you can find us over there, and we're pretty active on Instagram and Facebook. So with that, I just want to thank everybody for giving me the opportunity to talk about some of our work, and be happy to answer any questions if there's time. I know I went about 4 minutes over, so apologies for that.
Yes, thank you, Nate. So we will have question and answer session after another talk from Chuck, and there might be some questions in the chat box too. So our next presentation is, thank you, Nate. Our next presentation is from Chuck Chimera, and he's from Hawaii Invasive Species Council, and he has done lots of work in weed risk assessment and some of the prevention policy of weed. Chuck.
Thanks,
Great.
Roshan. Can you guys see my presentation?
Yes, but
Okay.
not full screen. Yeah,
Full screen now?
yeah, full screen.
All right, great. Thank you very much. Thanks for those of you who are sticking around to the bitter end. As Roshan said, my name is Chuck Chimera. I am the Hawaii Weed Risk Assessment Specialist for the state, and until they perfect the WRA bot, I will continue to be the human end of the weed risk assessment process. This project is funded by the Hawaii Invasive Species Council under the University of Hawaii Pacific Cooperative Studies Unit. So today I'm going to talk about flagship pest plants and their probable pathways. I picked the most alliterative title so that I would mess up right at the start. But before I continue, I'm going to focus on plants for my talk. I did want to acknowledge a large group of people that are part of this work and working on a broad variety of taxonomic groups. So not just plants. As part of this Pathways Task Working Group, identifying invasive pests across taxonomic groupings. And so I apologize if I left anybody off this list. These were all grabbed from emails shared through the various meetings we've had over the past few months. Again, I apologize if I forgot anyone, but I did want to mention specifically the leadership of Helen Sofer from USGS and Daniel Froelich from SWCA for kind of spearheading this task group and keeping us on target and making, forcing us to attend meetings when we didn't want to. But everybody on this list and on this slide has contributed. So I did want to acknowledge them all up front. I'll just be focusing on my area, which is plants. So this Pathways project that I'm going to talk about, I wanted to give a little bit of background about its origins and why we're doing this work. And so in the beginning, or actually not too long ago, there was the publication of this document. And if you're not familiar with it, the Hawaii Interagency Biosecurity Plan, it's a compilation of the state's needs in regard to invasive species and biosecurity across a whole broad area, from agriculture to the environment to human health. It's a really informative aspirational document that kind of lays the groundwork for where we as a state want to be in terms of biosecurity and invasive species management in the state. So if you're not familiar with this document and you haven't had a chance to look at it, it's fairly user-friendly, lots of colorful graphics in it, and you can download it at the link I've listed here. This document focuses very broadly on 4 program areas, which could be categorized as pre-border, border, post-border, and public awareness. So pre-border is the focus of this talk today. It's like, what do you do to prevent new invasive species from arriving into an area? And to delve a little deeper into this project that I'm going to be talking about today, the Hawaii Invasive Species Council and CGAPS, we heard the talk from Christy earlier, they put together this also this really nice document document, a 2025 joint strategy on how to implement and reach some of the goals laid out in the Hawaii Biosecurity Plan. This document can also be downloaded at the link I listed there. So if you haven't had a chance to look at this, you can see some of the goals that the state has set out to achieve our biosecurity future. But very broadly, this joint strategy lays out 10 priority strategies that we would like to achieve as a state to achieve maximum biosecurity, if you will. And the topic I'm talking today focuses on strategy number 1, which is prevention, early detection, and rapid response of terrestrial organisms. And in this joint strategy document, it identifies, or it describes this first strategy as keeping Hawaii's top invasive species threats from arriving and establishing in the state by identifying and addressing high-risk pathways in major modes of introduction, and by approving early detection and rapid response. And so this is what we're working towards with the project I'm going to be talking about. Now, within that same document, under strategy number 1, there are 13 key actions that are listed. And please don't strain your eyesight trying to read those. They're listed over there on the right. But the one I'm going to be focusing on is key action number 1 under this prevention strategy. And it is that the Hawaii Department of Agriculture, HISC, and other partners will collaborate on generating a non-regulatory Hawaii prevention priority list to guide our prevention and rapid response in the state. So this is the key action we're working towards. And to achieve this strategy and work on this key action, the first thing was to generate this prevention priority list. And so with that goal in mind, Kevin Hoffman, who's now retired head of HDOA, took the first crack at creating this prevention priority list. And the organisms on this list were broadly grouped into these categories I list below. So viruses, pathogens, and fungi, aquatic species, terrestrial plants, terrestrial invertebrates, and other organisms like snakes and other assorted land vertebrates. So Kevin Hoffman did a heroic job at taking a first crack at this list, and he distributed this early version in May of 2020 of last year. I think that was the first time I ever saw it. So we have our list in hand now, and so the next step was to collaborate on generating this list with all the partners. So the next step was the early detection rapid response groups met and formed a pathways task group to specifically focus on refining the list that Kevin produced. And as we are all aware, and as we're experiencing right now, this work took place during the era of COVID and Zoom meetings. So all of this work was conducted by various peoples across the state. And I took a screen grab of our working group, all happy and engaged in generating this list. And this is me in the middle here, cracking one of my many jokes. And so we had our task group, and now our work was to take this list that Kevin gave us and refine it for the next phase of the project. So at this point, I know Christy gave a really good overview of invasive species and how they arrive in Hawaii, but I just wanted to make sure that I'm going to be saying pathway, that if you're not familiar with this term, it's very commonly used in invasive species work, and this definition is taken directly from the Biosecurity Plan. It's just very simply the means by which an invasive species is transported from one location to another. So when I'm talking about pathways, that's what I mean. And this pathway definition can be divided and parseced into intentional, unintentional, accidental introduction, but pathway is just very simply this definition. So this pathways task group working on refining this list of species produced by Kevin Hoffman, as we were going through the list and meeting over the course of several months, the focus of this project started to shift the emphasis on flagship species, flagship invasive species, and the pathways by which they would most likely arrive in Hawaii. And some of those earlier documents, the biosecurity plan and the joint strategy, identify these species as sentinel species. So these are representative species that, should they arrive, they would kind of be an indicator of how good we were at addressing the particular pathway that they were associated with. So we had these list of prevention species that Kevin Hoffman produced, and we wanted to assign them pathways by which they would arrive. And we came up with this categorization process that was produced by the Convention of Biological Diversity, where they take pathways by which invasive species arrive and divide them into intentional, unintentional, and subcategories under each of those groupings. And so this is an internationally recognized standard of invasive species pathways, and we decided it would be a pretty good standard to adopt for Hawaii as well. I do want to emphasize that this work is still in progress. So if I mention any particular taxa today, I want to say that this list has not been finalized. It's still being refined and tweaked, getting input from various experts in the different taxonomic groups. We share all this information on a Google Drive, Google spreadsheets and documents. And, so it is very much a living document that's still to be finalized. But of those 43 conventional and biological diversity pathways that were mentioned in the previous document, we basically determined that about 10 of these are not really applicable for Hawaii. There's a bunch that are more applicable to continental or contiguous land masses, for example, tunnels and land bridges. So I haven't looked at the Biden infrastructure bill that was just passed yet. There may be a channel from the mainland to Hawaii in there, or a land bridge, and this pathway could be important in the future, but for now, 10 of these were kind of disregarded because it's unlikely any species would arrive on those pathways. That leaves 33 other pathways by which invasive species could arrive, and 9 of these are relevant to plants. So those are the ones I'm going to go into in more detail. And I'm going to be using this CBD and abbreviation quite a bit through my talk. So if you get excited, just want to emphasize I'm talking about the Convention on Biological Diversity, and not perhaps a more famous CBD you may have heard of. This could be a topic for a future talk, perhaps. So flagship species and their pathways. I'm going to focus on the plants, and Christy Martin already went into this in great detail, so I don't have to cover this too much, but I do want to say that the plants included on this list, the origins of these that Kevin Hoffman produced, were derived from that work that Shaheen Ansari had done, as Christy had mentioned, this offshore and incipient plant list, where she looked at invasive plants from around the world that were not present in Hawaii, and that were also present in trade, either commercial or private trade, and therefore had some likelihood of being introduced or arriving in Hawaii. So that's the list that Kevin worked off of and that Shaheen produced. And so that's the one that we proceeded with. And as Christy mentioned, there was 36 plants identified by Shaheen that were included on this prevention priority list. And we started assigning them pathways by which they might arrive. And all of these are essentially grouped under movement of a commodity. And the categories that are applicable to plants are in particular release in nature, escape from confinement, and transport contaminant. And under those 3 more general categories, there are several subcategories. And this table to the left is a listing of all the different CBD categories listed in the Convention of Biological Diversity. So release in nature includes introductions for erosion control or conservation purposes. Under escape from confinement, introductions for agriculture, aquaculture, botanical gardens, forestry, and horticulture are the subcategories. and as a transport contaminant, contaminant on animals, or seed contaminant. So now I'm going to go into the these different subcategories and the flagship or sentinel plants that have been assigned to each of these pathways, and just discuss why they were chosen. So the first category, release in nature, has a subcategory, introductions, a pathway for erosion control or dune stabilization. And for this subcategory, Rhizophora stylosa, or stilted mangrove, is the preliminary flagship species we've assigned to this category. The WRA for this plant is still in progress, but it is invasive elsewhere. It's known to form dense thickets in coastal habitats and estuaries, which is pretty common of a lot of mangroves around the world. That's what they do. And like the related red mangrove, which is already established in the Hawaiian Islands, it can also invade fish ponds. So in addition to biological and environmental impacts, it could impact Hawaiian cultural practices here. So we know it's invasive. We know Hawaii doesn't have any native mangroves, but we also know that mangrove conservation and cultivation is being discussed as a possible mitigation measure for climate change, sea level related climate change, especially where mangroves are native. Preservation of mangrove habitat is considered important to protect coastal habitats, protect from storm surge, sea level rise, and interest in that kind of use could increase in Hawaii as the impacts of climate change are felt in the future. So this is a pathway that could be important for future plant introductions. The next category under release in nature, the next subcategory, is introduction for conservation purposes or wildlife management. This may not be a super important pathway for Hawaii, but it's still a way that invasive plants could arrive. And for this subcategory, the flagship species that we've chosen for now is Gomphocarpus fruticosus, or swan milkweed. It received a very high WRA score of 27. It's got a lot of invasive traits. It is toxic, and it is documented to be an agricultural and an environmental weed where it's introduced. But like a lot of native and non-native milkweed species, it is also known to be a host of the monarch butterfly caterpillar or larvae. And on the mainland, there's a lot of interest in monarch butterfly conservation. People are encouraged to plant butterfly gardens, plant milkweed for the monarch butterflies that are migrating between North America and Mexico. And so some of that good intention could spill over into Hawaii, where people want to help. We have introduced monarch butterflies here. They're not native, but some well-meaning individual could decide they want to help monarch butterflies by introducing an invasive plant that's not here yet. We already have invasive milkweeds here, as well as crown flower that also hosts the monarch butterfly. And we would encourage people to plant those rather than introduce something new. But again, this is a flagship pest, or flagship plant meant to represent any plant that could be introduced for conservation purposes. So the next bigger grouping, escape from confinement. This is where something is cultivated but isn't intentionally released into the wild. And obviously agriculture is a very important pathway of introduction everywhere in the world, including the Hawaiian Islands. So for this subcategory of agriculture, the flagship species chosen is Rubus fruticosus, or European blackberry. This is really meant to be more of like a surrogate species for all non-native rubus that have yet to be introduced to the Hawaiian Islands. We have several introduced rubus species here, as well as 2 native species. And I think almost all of them have become invasive. This species has has also received a high risk score with the WRA, and it has become an agricultural and environmental weed where introduced. The spiny, thorny rubus species can become quite an agricultural pest in ranches and in grasslands. They can be spread by animals and birds and other animals that eat their fruit, and they can also spread vegetatively. So Rubus are really good at becoming invasive where introduced, and but they're also widely grown. Everybody loves blackberries and raspberries, so a lot of interest in cultivating them. And again, this is a flagship species meant to represent all potential invasive plants introduced through the agriculture pathway. The next category under escape from confinement, kind of almost like a subset of agriculture, is aquaculture. And the WRA for this species is in progress, but the flagship under this subcategory is currently Trapa natans, or water chestnut. This is a non-native plant that grows in very dense mats that can displace native vegetation and affect water quality. It's very invasive in the continental US. These spiny nuts pictured over there on the left can also cause injury if you're trying to swim or navigate through them. But this is the water chestnut that is used in a lot of cuisines, and it is cultivated globally for its nutritious nut. So, almost all freshwater aquatic plants that get introduced out of their native range become very invasive in their new habitats. So we do have several aquatic invasive plants already present in Hawaii, and this is another pathway to introduce new ones. Under escape from confinement, another subcategory, a pathway could be introduction through botanical gardens, zoo, and aquaria. And so for plants, obviously, this would be botanical gardens. And for this subcategory, the plant that was chosen as the flagship is Melianthus camosus, or tufted honeyflower. This is a weed of pasture land that also invades riparian habitats in Australia. This is a native of South Africa, and it is able to form dense thickets that can almost eliminate all other plant species. And this was chosen to represent the botanical garden pathway, not because this species in particular would be introduced in a botanical garden, but hypothetically, any species could be introduced through a botanical garden, if there was some kind of cultivation or conservation or scientific interest. And this has been an important pathway for introductions of invasive plants in the past. But fortunately, almost all of our major botanical gardens in Hawaii right now are very proactive at identifying and managing plants that have a potential for invasiveness now. So it's still a pathway for new invasive species to arrive in Hawaii, but I don't think it's like the major one today that we have to be concerned about. But it is still a way new invasive plants could reach our islands. Oh, and I did, in reading about this plant, I did want to mention there was some interesting information I read about it. This is not a CBD pathway, jackal trap masking, but I did find in researching this plant that it's very, I guess, has a very strong, unpleasant smell. And in its native South Africa, it's used to wipe the ground and remove the smell of humans when placing traps for jackals and other wild animals. So if we fail at our job and jackals somehow get introduced into Hawaii in the future, and then we have to start trapping them, this could be an important pathway for introduction of the tufted honeyflower. But right now, probably an unlikely way this plant would be introduced. The next category, or the next subcategory under escape from confinement, and an important category for past invasive plant introductions, was through the forestry pathway. And for this subcategory, Cecropia schreiberiana, or trumpet tree, was chosen. This plant receives a high-risk score of 9. It's known to be invasive in Puerto Rico, where it forms dense, almost monospecific stands, and its seeds can be dispersed by birds and other fruit-eating animals, like fruit-eating bats, which we don't have in Hawaii. But it is invasive, and like its related Cecropia peltata, which is established in Hawaii, it would probably establish in a lot of similar habitats. But it does have uses, forestry uses. It's a pioneer species, so in some places it's being encouraged for use, especially in its native habitat, for reforestation. It's a fast-growing, early colonizing species, which is great in areas where it's native for reforestation purposes, but it's also one of the traits that's identified with a lot of our most invasive plants. It's also got a lot of uses. It's a fairly light wood, so probably not so much construction, but there's a whole bunch of other uses listed there. And that's the thing about forestry. It could be, could be any use that could be identified as a reason for introduction. So this continues to be an important pathway for potential invasive plant introductions in Hawaii. And probably, the major pathway for introduction of new plants into Hawaii is the horticulture and ornamental pathway. So for this category, subcategory of escape from confinement, the plant Myoporum latum, or Ngaio, or mousehole tree, was chosen as the flagship. This plant receives a high-risk score of 12 from the WRA. It does form dense monocultures and increases fire risk where it's invasive, especially in California on the mainland. This plant is native to New Zealand. It's also a host plant for the Nile thrips, an invasive insect pest that is already impacting our native Nile tree, our native Myoporum. And as far as we know, this plant has not been detected in Hawaii. It's kind of hard to believe that it's not already here, but at least it's not officially documented. But it could arrive because it is used as an ornamental plant for screens and hedges. It could even be mistaken. It superficially looks like our native Nile plant. So people could even think that they're growing Nile and perhaps growing this instead. But there are so many other invasive horticultural and ornamental plants that could be introduced. It's hard to choose just one. So we went with this one because it's also a host of a pest that's harming one of our native species. Now, the last category I wanted to talk about was transport contaminants. And the subcategory under this that's relevant to plants is a contaminant on animals. And I put a question mark there because I'm not really sure how important this is for introduction of new invasive plants into Hawaii today, but in the past, it could have been important. And it was certainly how, much of our native flora arrived in Hawaii, hitchhiking on birds and things that arrived here. But today, it's really a long shot that something would arrive hitchhiking on an animal, but it's still possible. So we're identifying it here. And for this category, the flagship chosen right now is Drymeria arenarioides, or Alpha umbrella. The WRA for this one is in progress, but this one's already listed as a federal noxious weed. It is regulated. It's also fatally poisonous to livestock. It's native to Mexico, where it invades, or I should say it's naturally occurring in rangelands there, but it is toxic to livestock, grazing animals, and they're really concerned about it spreading into adjacent lands in the United States and the southwestern US. That's why it was listed as a federal noxious weed. And it can be dispersed. It's got very small seeds, as you see there, that can be spread by wind, water, animal droppings, and also attached to animal hair. So if a cattle, if livestock were shipped from the mainland, I don't know how often or if that even occurs nowadays, but if that did happen, it's potential that a seed could perhaps be sticking to one of them. Or, people bring their pets to the islands from the mainland, perhaps like some invasive plant seed could be stuck to the animal fur. So this is a pathway that we have to be aware of. Again, I don't know how important it is today compared to the other pathways, but it's still one that we need to be aware of. And then the last subcategory under transport contaminant is seed contaminant. And for this subcategory, the flagship species is Anoportum acaulon, or stemless thistle. This plant received a high risk score of 10 with the weed risk assessment. It's also listed as a federal noxious weed. So it's already, a known invasive species elsewhere. It is regulated. And where it is invasive, it reduces the carrying capacity of grazing land, and it can be injurious to wildlife because, like a lot of thistles that you may be familiar with, it's covered in these prickly leaves and very spiny inflorescences that can harm grazing animals as it invades their pasture. The seeds are spread by wind, animals, vehicles, but also as a contaminant of hay, silage, chaff, and commercial seeds. So, you know, this plant isn't obviously intentionally cultivated anywhere, but where it's growing adjacent to, you know, commercial crops or agricultural pastures, the wind-blown seeds could be blown in and then contaminate the seeds of these adjacent areas. So, but there's so many other invasive plants that could be introduced as a seed contaminant, and it is a regulated pathway. But again, this is our flagship that we've chosen to identify to highlight this subcategory. So I'm almost done here. Why is this important? Well, Christy again gave a really good overview of all the different ways we've tried to prevent the introduction of new invasive species, and particularly invasive plants in Hawaii. But if you may have noticed, of all the different subcategories I was addressing for plants, with the exception of the last two, all the plants are essentially allowed into the state through intentional introductions. These are intentional pathways. And another way of putting that is that very few plants are legally prohibited from entry into Hawaii. You know, Christy mentioned the federal and state noxious weed and seed rules. So that's a few hundred species that are legally prohibited from entry into the state. We also have that restricted plant list. You may, I think Christy also mentioned the study, this publication that Cliff Smith, the retired UH botany professor, did in 1985. It's often studied. He looked at weeds of natural areas that were impacting, you know, like national parks, state natural area reserves, you know, more of our native dominated habitat. And of the species that he identified as the worst invaders in these areas, almost, you know, more than 90% of them were intentionally introduced. So they were brought in for one of these other reasons that I mentioned, you know, ornamental, forestry, some other agricultural use. Very few, you know, less than 10% were accidentally introduced. So this is just a way to emphasize that we really do need to update and revise our noxious weed and restricted plant rules, you know, as soon as we possibly can. And there are different working groups also have met to try to do that. This isn't a self plug for myself, but, you know, barring or until we address those, you know, until we update our rules, we should really be screening all new plant introductions for invasiveness before they are introduced or widely cultivated. And the next steps in this project, as I mentioned, this is work in progress. So we're going to be finalizing the lists of the different taxonomic groups. So, you know, in addition to the plants I discussed, there's all the pathogens and invertebrates, aquatic species that are being assigned pathways. So we're going to continue to meet to decide on which species best, you know, best represents the flagship species for that pathway. Once this list is finalized, again, it's non-regulatory at this point, so it's going to be used as a promotion means to promote outreach, awareness, and possibly be used as a tool for future legislation and regulation. And in one of my earlier slides, I mentioned all the key actions, the 13 key actions for that first strategy, prevention strategy. And one of the other key actions on that, in that document, was to track and report annually on the status of all these prevention list species. So determine which ones are not established every year, which ones have been detected and intercepted or prevented before introduction, or, you know, if we've failed at our job, which ones have become established. There's also a project proposal that's possibly going to be, or hopefully going to be funded soon or in the future, to do what's called horizon scanning, which is a much more formal and rigorous evaluation of which invasive organisms are likely to arrive, establish, and impact an area, in this case, Hawaii. And if any of you are members of the North American Invasive Species Management Organization, a really excellent talk was given just yesterday morning by Dr. Wesley Daniel on this horizon scanning process for organisms in trade. So invasive animals that are in the pet or animal trade that pose a risk if introduced into the United States. And so this analysis included Hawaii as one of the regions at risk. So it was a really, really informative talk. If you want to learn more about horizon scanning, I would suggest visiting this website, and you know, they might be posting a copy of this talk in the future. And then we have all these other strategies and you know, key actions to work on. And so after the prevention list is finalized, we can work on the limited distribution priority list, which is species that are already present in Hawaii, but are not yet widespread. So how can we prevent them from expanding the range or being introduced to other regions or islands where they're not present? And then once we've done that, we can satisfy ourselves with a job well done, and with all the, you know, protection we've provided to the state and all the invasive species we've solved, and we can rest on our laurels. So with that, I'm finished, but I'm happy to answer any questions if there is still time. And thank you for your time.
Thank you, Chuck. I think we have some questions in the chat box. Let me see. So this is question for Nate. So it says, already seeing Myconia replacing rot killed Ohia in Puna on. So another question actually. So it says Myconia is replacing rot killed Ohia in Puna and Big Island. Is that correct? So
I would guess so. Yeah.
Mhm,
I mean, I don't know specific. Somebody from BIS could probably speak more to it, but there's a lot of Myconia on Big Island, and there's a lot of rapid Ohia death on Big Island. So that would make sense.
So Nate, I have a question. What is difference between young Myconia and mature Myconia? The mature means it's already flowering, or it's producing seeds?
Yes.
So how big
fruiting
is the
or
seed?
flowering.
How big is the seed?
They're about the size of a granule of sand. They're very small. The fruits are small and they contain a lot of seeds.
Okay, so that means it can spread rapidly. Okay, thank you. So in your survey, so you just all the maps you show after 5 years or 7 years or 10 years, is that the simulation based on the?
Yeah, based on the parameters that we draw from the species information that we have, and based on information about dispersal or the vectors that disperse the seeds. We input that all into our model, and then it takes a lot of computing. And one of the reasons we keep it conservative is because it's a lot of computing for the model to put out all this data, all these projections. But it's also helpful to keep it conservative because you could see just how much it was blanketing those areas, even at a very conservative estimate. So it does tell a little bit more of a distinct story to the people we're trying to get funding for or access to certain areas or things. But yeah, basically it's a predictor of what could potentially happen if we don't continue with the important work that OISC has been doing for the last 2 decades.
So if you put your control methods of the number of number of Myconia killed also, would that be the different output? Can you put that parameter, that how many trees you will be killing in each year?
Well, we can't really predict that because it just depends on where we find the plants. So we can't predict it through the model. We do take that into consideration when we're telling the narrative, let's say in a proposal to acquire funding or in a proposal to gain access to a new area. But basically what the model is being used mostly for is to say, if we stopped doing our effort because we don't have enough funding or because any other factors, then this is what would happen with Myconia in these areas. So when we first started, it was kind of, we just did it from year kind of 0 in terms of these are all the plants that we found the first year. If OS didn't go in and survey and control like they did, this is likely what would have happened. So those were kind of those first images. And then since then, we use the model more frequently now. And we take data, like for example, in Kalaya, we had controlled, I think it was 2 mature and 206 immature plants so far that year. But we're looking for more funding because that's one of our higher priority watersheds, one of the denser populated watersheds with Myconia. So we put that into a proposal and say, we found this many this many mature trees and this many immature trees, and we plug that into the model. The model spits out the maps and the figures that I shared with you folks today. And then we transfer that over to a proposal and say, we're going to run out of funding. We need more funding. Please give us more funding, or this is going to be the result. So it's a different kind of way of explaining the story, but it does get a little tricky in trying to get a funding for invasive species. So trying to incorporate these new tools, it really helps us tell a better story, a more diverse narrative. And yeah, so hopefully that answers your question.
Yes, thank you. So there's one long question. So I don't want to read this. Maybe if anybody can address this question, the one long question. I think she had a couple of those. Okay, so forget about this. There's I think this question is also for Nate. Are these partners? No. Are these partners that can help with control in the wildland summit areas? Wildland. So.
Our partner organizations, yeah, we do have a broad network of partners who support us in a variety of ways, whether that's funding or with collaborative projects. We focus on Myconia because it's kind of our bread and butter, and it allows us to keep it off the plate of other agencies who deal more specific to regions. So for example, a mountain watershed partnership really is focused on a certain area. They have boundaries and they have a variety of different work, whether that's invasive species control or outplanting or fencing or fence monitoring. OISC doesn't have any set area for any set species. We follow the species and we follow it across land ownerships. So as you can expect on Oahu, we have tens of thousands of different properties that we have to deal with, and thousands of properties each year that we deal with. And so because we're not specific to any one particular place and we follow the species, I guess that allows us to better utilize, well, I guess we're just not restricted to those areas. So yeah, anyway, I'm not sure if I might have lost your question, but
No problem.
if you can better explain it, I'd be happy to jump back in.
Anyway, thank you. So there is some information from Christy. Thank you, Christy. Then again, there is a question for myoporium trips. I think James Friday answered this. And we have a question. Is the Myconia biocontrol status? Do we have information on Myconia biocontrol?
That's I can jump in real fast for that. It's still underway. The process is still underway. I don't know the specifics, but Christy or Chuck may be able to speak a little bit more directly to the biocontrol agent specifically. But as I understand it, it has not been released yet, but it is in the process of the lengthy biocontrol process.
I did put the link in the chat. Sorry for just chiming in, but Forrest and Kim clarified about how about the Colototricum on Oahu.
So I'm sorry, Christy, the link for Myconia biocontrol?
Yes.
Okay,
Yeah,
thank
and
you.
right below that, they clarified that it was the Colototricum that they were asking about. From my limited field experience, I have not noticed it obviously on Oahu Myconia.
Okay, thank you.
Yeah, our field crews haven't reported any, they're aware, but they have not reported any sightings either.
Do we know if it's working on the Big Island?
Yeah, there's no Myconia here anymore. All taken care of.
You're the best, Chuck.
No, I'm not aware of any work, evaluating its effectiveness personally. But I did just put in the chat. You'd think I was a paid representative for NASMA, the North American Invasive Species Management Association, but I'm all about free stuff. And I think it's next week they're going to be hosting a biocontrol summit, which I think is free. And I saw that, two of the speakers at the summit are going to be Tracy Johnson, our U.S. Forest Service biocontrol expert, and Darcy Oishi. And I saw that, among the species, Tracy will be discussing our Myconia and, a bunch of our other favorite invasive weeds. So like I said, I think it's free. So he'll be giving, probably a pretty up-to-date review of where we are right now with that.
Did you put the link in the chat? Thanks, Chuck.
Okay, thank you. So I will just move quickly through. How about the fungus Coleotrichum for Myconia? Does anybody have information on fungus? This is for Myconia biocontrol, right?
Yeah, it's still found on leaf surfaces in Hana, on Maui. And so someone from, I think it was from USDA, was just about to do rearing of it, and they lost their funding.
Yeah, this was a fungus that was released many years ago, and it's like a little light, little surface spots on the leaves.
Okay, yeah. Okay, next question. This one is for Chuck. How might climate change affect the pathways by which invasive plants may enter, become problematic in Hawaii? And could climate change affect which species are the highest invasive risk in Hawaii? Chuck.
How could climate change? Absolutely, climate change could affect pathways. I would say there's a couple of ways that it could affect. I mean, the species that are already established here could start, shifting their ranges, perhaps moving up slope as temperatures increase or as precipitation patterns change. Certain areas become drier. Invasive species that are already here, like fountain grass or other fire-loving species, are going to spread into areas that become, more arid. I know that maybe some of the climate models predict, stronger rains or more torrential rainfall, but over a shorter period of time. So, I'm certain that, invasive species, so many of them are kind of pioneers that, exploit disturbance. So, disturbance related to, frequent, more intense storms are certainly going to benefit the species that are already here. As far as like introduction of new species, I mean, as our climate gets warmer, I suppose there could be, one of the pathways could be, climate change mitigation for maybe like carbon sequestration or something, that we want to plant something to offset climate, carbon footprint somewhere else. So that's indirectly related to climate. As far as like, something being introduced related to climate change, yeah, I'd say that, as the climates, the precipitation, temperatures change, that's going to change what can grow in different areas. So that will, increase and perhaps expand interest in things that we don't currently have here. So it's kind of like a very broad generalization, but absolutely, I think it'll continue to affect what can arrive and establish.
Thank you, Chuck. Chuck, I have a question. So suppose if I would bring my favorite blackgram varieties from my home country and grow in my backyard, what would be the pathway? Is that
If you were to bring a plant, well, I mean,
seed?
the seed, I would say the pathway would be, intentional introduction through horticulture or ornamental, depending on what you're using it for, agricultural purposes, which is all legal. I mean, as long as it's not,
legal,
one of those few prohibited plant species. I mean, we could get online and start ordering seeds from pretty much anywhere in the world. I forgot to mention that almost every plant I mentioned,
next
has
question is, would it be
availability of
considered
seeds.
as an invasive plant?
Is what it?
Would it be considered as an invasive plant? My blackgram, my favorite variety of blackgram, I would grow in my backyard.
I mean, it depends on, its behavior elsewhere. If it's identified as invasive elsewhere in an area, similar region, similar climate, or areas where it's been introduced. I mean, one of the best predictors of whether or not something is going to be invasive here is if it's already got a history of invasiveness elsewhere. If it doesn't, it still may share biological traits, with some of our worst invasive species. So how it's dispersed, how it reproduces, what its climate tolerances are, those are all considerations. But the best predictor is if it's invasive elsewhere.
Okay, thank you. So another question, I just became aware of possible Penicetum pilosum planting in a display garden at upcountry display. So can you look at this question? I don't know if it is. I just became aware of this.
Is that one for me or for somebody else?
It's somebody else.
Okay.
It's for Chuck.
Chuck, yes, okay.
Hey, Mike.
Hey, Chuck.
Okay, Mike.
Yeah, so I just had a report of it with image from, upcountry in a display garden. So I don't know. Is it? I wasn't sure what to do. I just saw it on your list. I was like, oh my goodness, it's not here. So anyway,
Which plant was it?
Velocum, Centris, or Penicetum. So it was on that list that you just put up. So and not known to be here. But,
So we've already failed. No, I also forgot to mention that, one of the key criteria of these species we selected is that they're not known to be present in Hawaii. And that's only based on the best information we currently have available. So if anybody, knows of any of the plants I talked about already being present in Hawaii, we'll put something else in as that pathway. But because this is non-regulatory, it's meant more for educational purposes at this stage. That could change in the future, hopefully. But for now, it's just to emphasize all the different pathways where invasive plants and other organisms can arrive in Hawaii. So Velocum, yeah, I mean, like somebody could go online right now, please don't do this, and order seeds from all the plants that I just identified. And you could basically destroy months of work in a few mouse clicks. So, but I mean, seriously, that was my take-home message, is that all these pathways are basically legal methods for introducing plants into Hawaii, and will continue to be. We're one of 50 states. We're a relatively small economy, and relative to some other areas. And I'm not going to get on a soapbox, but people can privatize their gain, and then if it becomes a problem, we socialize the cost. So we all pay for it in the long run. And so we're just trying to do the best we can to continue to raise awareness that we are inviting these things here, in many cases intentionally, and then dealing with the fallout sometimes much later.
So for Penicetum Velocum, we remember many years ago, the late, rest in peace, Lloyd Loop was shopping at Kmart, rest in peace, and came across Penicillin villosum seed packets, Martha Stewart branded Penicillin villosum. So it's you know, yeah, the more that we know which one of these are the ones that we should really be reporting, the better, because there's a lot of stuff happening all the time. And yeah, we can do a better job of staying on top of it. And we appreciate all your work with this, Chuck. You know, your HWRA, it's really, it's a great thing to be able to point people to, and just help us kind of just get a framework for all of this. Mahalo.
Group effort.
Thank you. So then there is some information on biocontrol summit from Chuck as well as Chelsea. Thank you. And Chuck, can you look at the chat in the chat box from James, JB Friday?
Oh, I see. It said we
Yeah.
definitely have had groups want to
Mhm.
come here and plant non-native and potentially invasive trees to absorb CO2. Yep. Not surprising at all. I mean, you know, and I'm not even, you know, saying, you know, black and white, that isn't something that we should be doing here. You know, I mean, it's all a cost benefit analysis. I mean, you know, even with mangroves, which, you know, are not native to Hawaii, I mean, maybe the pressure to protect coastal areas may outweigh any other consideration, you know, and that's something that, you know, we as a state and as a community, hopefully will decide together, and the decision won't be made, you know, for us by, you know, one special interest. But yeah, I mean, I know that, you know, we're always introducing new species, many of them that have the potential to become invasive, and you know, that's the situation that will probably continue to happen in the future. But we're doing what we can to raise awareness about it.
Thanks, Chuck. So there is another question. How are we helping the public know about avoiding these plants?
Well, as Christy touched upon in her talk, and I will promote again, you know, the a plant website that I'm affiliated with, where the weed risk assessment results get posted, is Plant Pono, and Molly Murphy from the Big Island Invasive Species Committee on this island, on Big Island, is very proactive at engaging, as well as the whole BISC outreach staff, is really proactive at engaging nurseries, growers, plant people. They're in you know, when we could meet in person, they would have, like, you know, outreach events. Molly goes and visits nurseries. She visits, you know, people selling plants. She engages with them online. She's much nicer than me, so they don't get angry and scared away. But I know that every island has outreach people in the ISCs. They're all excellent. They all do a great job. They're all constantly, you know, raising awareness and trying to, you know, encourage people to, you know, avoid invasive species and, you know, and help. But yeah, we can, as much as we're doing, we could always do more.
Okay, thank you. So just a last minute question. One last question can ask in person, not in the chat box, if anybody has. Then we can conclude. So it looks like we don't have any more questions. So thank you, speakers. Thank you, Chuck. Thank you, Nate, Christy, and Ryan. I'm very thankful to you and all the participants who we hang out until this time, around already lunchtime. So with that, I thank you all, and I will see you in another couple of months. Thank you, everyone.
Thanks,
Thank you
Roshan.
all.
Bye-bye.
Thanks for putting this together.
Good afternoon.
Aloha.
Yeah,
Thanks,
mahalo.
everybody.
Yeah.